my group is doing at the Christina River Basin Critical Zone Observatory, which is located in southeastern Pennsylvania, northern Delaware, where we're looking at the cycling of carbon in the terrestrial environment. And this morning I want to particularly emphasize the role that carbon mineral complexation plays in carbon cycling and sequestration. I want to also acknowledge my co-author, Chun-Mei Chen, a graduate student in my group, and I'll be reporting on quite a bit of her research this morning. I'll start out by showing a diagram of the critical zone, which may be a term that you're not familiar with, but it's a term that's catching on around the world. And the critical zone is actually that part of planet Earth where we live. It extends from the tops of the trees. It includes the Earth's surface, lakes, streams, animals, plants, down into the soils, down into the vadose zone, into the groundwater. And one of the nice things about the critical zone is that it tries to describe things across a range of spatial and temporal scales. Spatial scales extending all the way from the atomic to the global, and temporal scales from seconds to eons. And it also tries to describe the coupled physical, chemical, and biological processes that occurs in the critical zone. So here you can see that with critical zone science, one looks from very small scale at the molecular, even down to the atomic, all the way up to landscape and basin scales. And critical zone observatories are increasing around the world. This shows the critical zone observatories in different parts of the world are increasing. We have a number in North America, a number in Europe, South America, growing in Asia. In the United States, we have 6 critical zone observatories that have been established. 3 more have recently been funded by the National Science Foundation. Our critical zone observatory is here, the Christina River Basin, CZO, which is located in northern Delaware and southeastern Pennsylvania. It's a very large watershed that we're looking at, where we're trying to understand the role of carbon, looking at carbon cycling and sequestration. And we all know that soils are extremely important reservoirs of carbon. They are very important sources of sequestering carbon. Here you can see the different soil orders, and in many cases, particularly some of these soil orders, can contain and sequester very large amounts of carbon. So what I'm going to talk about this morning is the role that organic matter mineral complexation plays in the cycling of carbon. And this is certainly one of the major mechanisms for carbon stabilization. And this sequestration is controlled certainly by an understanding of the chemistry of organic matter, but also the important role that mineralogy, in particular the iron, manganese, and aluminum oxides play, as well as the limonosilicate. Organic matter, I think all of us that are soil scientists know, is probably the most complex component of the soil. And while there have been numerous studies on the chemistry of organic matter, we still don't understand it very well, particularly the structure, because it's so heterogeneous. And here you can see a number of proposed mechanisms for organic matter mineral complexation. I'm sure that there are a range of these that occur in soils. So I'm going to talk a little bit about how we're trying to sort out what these particular mechanisms are. So in our particular research, this is our hypothesis that organo-mineral complexation is a key process for stabilizing carbon, and it is limited by the supply of the mineral surfaces and the mixing of the minerals with organic matter in natural ecosystems. And so if one looks at many types of relationships between organic carbon and surface area, one sees often a very strong correlation between surface area and organic carbon. This is a cartoon that just sort of tries to illustrate the important role that environmental redox interfaces play in the biogeochemical cycling of nutrients such as carbon, but also metals. And the metal oxides and hydroxides, for example, iron oxides, hydroxides, are extremely important because they generally have larger surface areas, and so they can sequester and bind carbon. At the same time, the iron and manganese oxides can undergo reductive dissolution, causing carbon to be released. So, and what I'm going to talk about quite a bit this morning is the role, particularly of iron oxides, oxyhydroxides, play in carbon mineral complexation. Again, this is a cartoon that just shows you that these reactions are very susceptible to the redox chemistry. For example, when we're dealing with oxic conditions, we have primarily the iron 3 oxides and phyllosilicates. Then, as redox becomes more negative, we become more anoxic through microbial or chemical reductions, then these can be reduced to iron 2 solids as well as a lot of other materials, and then through absorption and precipitation, and again through oxidation, we can go back to the iron 3 oxides and phyllosilicates. So the objectives of the talk this morning are, one, to investigate soil organic matter composition as it changes under varying landscape topographic positions, to try to get some insights into the interaction of carbon and carbon forms with minerals at a small scale, and to characterize iron speciation along redox gradients. This is just a cartoon that we have a number of sites where we have hill slopes where we go from very oxidized conditions in the uplands down into the low-lying foot slope areas. And in these areas, of course, things are not as well drained. We have anoxic conditions where we could get slow carbon decomposition due to lignin preservation. At the same time, it's under these conditions where we could get reductive dissolution of the iron oxides, causing DOC, or dissolved organic matter, being released. So it's a very dynamic system depending on the topographic position. So within our critical zone observatory, we're very interested in not only the landscape position, but the land use. How does land use affect carbon cycling? And so we have a forested site, we have an agricultural site, a pasture site, and a floodplain site. Now I'm going to introduce now some of the techniques that we have used to try to understand the mechanisms of this complexation. And we rely in our group a lot on synchrotron radiation. We all know from the electromagnetic spectrum that at large wavelengths we see large objects. And when these wavelengths become very small, we can see things like bacteria, proteins, water molecules, etc. And it's fortuitous that the binding energy of electrons and atoms and molecules and biological processes range from about 10 to 10,000 electron volts. And it is in this region, from the infrared to the hard X-rays, where we can employ these very intense light sources to look at things at very small scales. And so I'm going to talk this morning about the application of some of these techniques, including near-edge X-ray absorption fine structure spectroscopy. Our work has been conducted at the Canadian light source. This is on the SGM beamline. And here one can characterize nitrogen and carbon functional groups with very minimal pretreatment. You can see the spot sizes, so this is more of a bulk technique. And then if we wanted to go to smaller scales, we can go to spectral microscopy, where one would couple both scanning transmission X-ray microscopy with near-edge X-ray absorption fine structure spectroscopy. And here we can acquire images over very small resolutions of 30 to 40 nanometers. We can map distribution of carbon and understand how it's associated with different elements, and get some insights into the mechanisms of carbon complexation. We've also employed XS, extended X-ray absorption fine structure spectroscopy, to get more insights into iron speciation, and also precise technique for delineating the speciation of iron chemistry. And of course, we've used standard X-ray diffraction. So let me talk a little bit about some of the molecular scale data that have been collected. This is on the pasture soil, where we're looking at a soil clay fraction, where particulate organic matter has been removed. We've tried to minimize any kind of artifacts by sonifying the sample. And here we can look at the NEXAFS data to get some insights into organic functional group distribution. And what we see, depending on whether we're talking about the foot slope, the back slope, or the summit, that in the foot slope soil, where we have less well-drained conditions, we see an enrichment in aromatic carbon, a depletion in aliphatic carbon, and the carboxyl group is also less intense, which would be expected under these anoxic conditions. We can then use the STIXM, the scanning transmission X-ray microscopy, to get spatial resolution of these samples and look at correlation between elements. And here I'm showing you tricolor maps where we look, for example, one sees a very strong correlation between iron and aluminum, which is not unexpected because the iron oxides coat the aluminosilicates, and we also have structural iron in the phyllosilicates. You see these blue areas, which basically represent silicon dioxide quartz particles, showing that they're not very reactive, as we would expect. There's a very strong correlation between carbon and calcium, which has been noted before. Through our XAFS analysis, we don't pick up any inorganic carbon through calcium carbonate, so all the carbon is organic, and we see this very strong correlation with calcium, which could be related to simply electrostatic absorption of calcium on the organic matter, or it could be cation bridges. And clearly, there is a very strong correlation between carbon and iron. A little less correlation with aluminum. This shows you the actual correlation maps where we've taken optical density measurements and then translated those to thickness effects. And again, you see this very strong correlation in both the summit and the foot slope to calcium, also to iron, a little bit less with aluminum, and very low correlations with silicon. So clearly calcium and iron are very important in terms of the complexation with carbon. We can also do combination of STIXM and NEXAFS to, in this case, to look at distribution of organic matter using PCA and cluster analysis. Here we're looking at different regions of carbon. The blue would indicate the lack of any carbon. The other regions, the colored regions, you can see these various NEXAFS peaks. And not surprising, we see these major functional groups such as aromatic carbon, carboxylic carbon, and polysaccharides. So if we look a little closer at each particular element, this is the calcium, and the calcium is clearly associated with organic matter. These are the extracellular polymeric substances. Also with calcium phosphate, you don't see any kind of calcite present in the soil. With iron, the iron is primarily iron 3, as you can see from the NEXAFS data. These are the soil clay fractions. You see a very strong relationship with the iron 3, lack of any iron 2. And then we can try to support those data through XAFS, where again we look at the distribution of iron 3 in the clays and oxides. You can see we have iron 3 associated with vermiculite and illite. We have a lot of iron 3, some ferrohydrite, but a lot of crystalline goethite and hematite. With silicon, we have a combination of association with quartz as well as with clays like muscovite, for example. So clearly, the iron is playing a very important role in the complexation and the cycle of carbon. And so I want to now talk a little bit about some of the work that we've done this again is a cartoon that shows you one of the regions that we're looking at. You can see this hill slope area. We have a floodplain area here with the western and eastern floodplain. And some very interesting soils and mineralogy in these different samples. These post-colonial deposits, you can see, look fairly iron-rich. The pre-colonial deposits, clearly they're more anoxic. You can see the modeling that's present. And at this particular site, we have installed a number of in-situ real-time sensors where we can measure things like temperature, redox, and soil moisture. And then we can simply, over real time, look at the redox behavior in the field. And clearly here we see that these buried wetlands are pretty much, they're reduced over this whole time period of several months, whereas the post-colonial and the gravel, you can see we're dealing with more oxic conditions over the time period. So here we've looked at the correlation between iron by sodium dicitrate bicarbonate, and then looked at that in relationship to mineral surface area. And clearly we see that in these pre-colonial wetlands where we have more reductive dissolution of the iron oxides, that these are the iron is quite low, and we see a very much different behavior in surface area compared to these post-colonial deposits that are more oxic. So here we can clearly see the effect of reductive dissolution of the iron oxides on and the role of surface area. The mineralogy is, we see a combination of both 2-to-1 and 1-to-1 clays. We see this hydroxy interlayed vermiculite in the post-colonial, illite kaolinite, and then in the pre-colonial we see presence of chlorite, mica, and kaolinite. If we look at the iron chemistry using Mossbauer spectroscopy in these different sediments, we see very striking differences between the wetland area and the colonial deposits. In these deposits, it may be difficult to see here, but we see primarily iron 3 oxyhydroxides present, as well as structural iron 3 in the clays. In the wetland sample, we don't observe any kind of iron 3 oxyhydroxides. These have all been undergone reductive dissolution to the iron 2. And what this really also shows is that in addition to the redox affecting the reductive dissolution of the iron oxides, we also see changes in the structural iron in the clay minerals as a function of redox. The XAFS data backs up very nicely the Mossbauer data. You can see that in the pre-colonial wetlands we see, and post-colonial we see iron 3. Again, the iron 3 oxides are only present in the post-colonial deposits. We don't observe any iron 3 oxides in these wetland areas. So this is consistent with what was found through the Mossbauer spectroscopy. So the last part of the talk this morning, clearly we see this impact of iron oxides. And so we decided to try to understand at a more model system what might be happening with carbon complexation, particularly with ferrohydrite. Most of you know that ferrohydrite is an amorphous iron oxide. It's very common in the environment. It's nanoscale in many cases. Very small particle size, very high surface area, and highly reactive with organic matter. So here we wanted to investigate the extent of organic matter absorption and co-precipitation. So we're looking at both iron absorbed onto ferrohydrite, or organic matter, as well as co-precipitated samples. Secondly, we were interested in understanding what is the stability of carbon through both absorbed phase as well as a co-precipitated phase. Thirdly, to compare the mechanisms of organic matter ferrohydrite complexation formation via absorption and co-precipitation. And then to get some insights on the spatial distribution at the nanometer scale. So this was our experimental setup. These were all laboratory studies in absorption. We took dissolved organic matter from a forested litter layer. We simply mixed that with freshly synthesized ferrihydride at 2 pHs. In the co-precipitated samples, we dissolved ferric iron in DOM solution and raised the pH to 7. And then in order to get some insights into the stability of the carbon, we used a number of desorbing agents, such as sodium phosphate, sodium pyrophosphate, and sodium hydroxide. And then we did a number of characterization through Fourier transform infrared spectroscopy, NEXAF, STIXM, and iron NEXAFs. So here I'm showing you the absorption, under absorption and co-precipitation, the effect of initial carbon to iron ratios. And what you see is for both absorption and co-precipitation, up to about 1 ratio, 1 carbon to iron ratio, we don't really see any difference between absorbed versus co-precipitated phases. Above that, with co-precipitation, we tend to see a continued increase in the amount of carbon absorbed as the carbon to iron ratio increases. This probably is related to organic matter iron cation complexes, which I'll show you later in some of the spectroscopic data. We then looked at the effect of organic carbon through absorption and co-precipitation, the level of that as a function of surface area. These were done at multiple absorption, desorption experiments, different partial pressures of nitrogen, and what you see is this very precipitous drop in surface area as the amount of carbon loading occurs, which is not surprising because the carbon is basically binding to the structure, covering up the sites so that they're no longer reactive. And we see about a full amount of coverage of ferrihydride at up to about 0.97 milligrams of carbon per square meter. We also try to look at what is the effect upon the micropore and the mesopore volume as a function of the loading of carbon. And again, you can see a very sharp drop in the surface in the micropore volume as the amount of carbon loading increases. And this is undoubtedly, again, due to the blocking of sites. And even though the carbon probably doesn't enter into the micropores, it can block the micropore openings, causing the decrease in the surface area. With the mesopore, we also see this drop in surface area, not quite as striking as we saw earlier, but and with the co-precipitation, you see a much more enhanced effect on the surface area compared to the absorbed samples. If we look at the stability of the carbon, here we're looking at the release of the carbon as impacted by different desorbing agents. Overall, you see quite a bit of stability of the carbon. The sodium pyrophosphate and the sodium hydroxide tend to release the most carbon, but we still see significant amounts of carbon being retained. Again, showing the very strong complexation between carbon and ferrihydride. With the co-precipitated samples, the carbon seems to be more stabilized than if it were just adsorbed. Now, to get some insights on the actual mechanism of the complexation between carbon and ferrihydride, these are FTIR spectra, where we looked at particularly the impact upon the symmetric COH band. And what we see is as we load more and more carbon onto the ferrihydride surface, one sees that the wave number for this band tends to decrease. We get a larger decrease at the lower carbon loading. So this suggests some very strong binding of the carbon to the ferrihydride, perhaps through ligand exchange type mechanisms. And we also see the asymmetric COH band being reduced and a new band appearing. So all this suggests that we've got fairly strong complexation between the carbon and the ferrihydride. With the co-precipitated samples, we see very similar behavior. We see this decrease in the band, the wave number for the symmetric COH band, and NEXAF spectra, again, this tends to back up the FTIR data because one sees basically here the COH peak tends to decrease as the amount of carbon loads for both adsorption and in co-precipitation we see the same effect. However, here we've looked at an organic matter iron 3 complex, and you can see at this higher loading that we have a shift in the energy which looks very much like the organic matter iron 3. So what we think is happening is that these larger carbon loadings, we get less precipitation of the ferrohydrite, and we get more appearance of the organic matter carbon cation complex. And this is illustrated even better in the iron XF spectra, where we looked at what is the distribution of iron for these co-precipitates as we load more carbon. And you can see at the low loadings, all of the iron 3 has precipitated to ferrohydrite. But as the loading increases, we tend to see the appearance of these organic iron 3 cation complexes that are occurring, showing that not all of the ferrohydrite, the iron 3 has precipitated as ferrohydrite. And this could be the part of the carbon which probably is less stable than where we actually form a pure co-precipitate. If we look at the Sticksom distribution of the carbon on the ferrohydrite, this is looking at for adsorption. What we see is that at low loadings, we tend to see a more homogeneous distribution of the carbon on the ferrohydrite. So again, suggesting and backing up the data that we saw with the FTIR and the NEXAFs, that at the lower carbon loadings, the carbon is more stable than at the higher loadings, where we see a more heterogeneous distribution. And the same effect is seen for co-precipitation, a more homogeneous distribution, low loading, a less homogeneous distribution, higher loading. So this suggests that at these higher loadings where these iron 3 cation complexes are occurring with the organic matter, that they're less stable than when we form just a clearly co-precipitate. So in conclusion, we can say that the co-precipitates results in greater carbon retention and stability than adsorption. Organic matter tends to be more stabilized by mineral surfaces and small mineral pores at lower loadings. Ligand exchange appears to be a major mechanism for the complexation. And under co-precipitation, higher carbon loadings, we see the appearance of these organic matter iron 3 complexes. So with that, I would like to acknowledge our funding sources, particularly the National Science Foundation, the work that we did at the Canadian Light Source, the Environmental Molecular Sciences Laboratory, and the XAF's iron data were collected at the Stanford Synchrotron Radiation Light Source. And lastly, I'd like to acknowledge my group at the University of Delaware. One of the privileges I have of going and speaking is that I can talk about data that someone else collected. So I really want to acknowledge the work of my graduate students. So with that, I'll